All right, so here we are in the lab. We have our lab drawer. I'm going to explain a little bit about some of the glassware and other instrumentation and materials we use in the lab to give you a little bit more experience so that when you come to lab and work, you'll be ready, and also so that you'll be able to be more educated about laboratory procedure and equipment. Here we have beakers. These are different sized beakers. Beakers have the same width at the base as they do at the height. We have graduates on the beakers, meaning these lines which indicate what volume is in the beakers. Because of the wide width of a beaker, generally these volumes are not very accurate or precise. So if I measure out 50 milliliters, I will not expect to actually have 50 milliliters. This is a 250 milliliter beaker, for example. The highest graduate it has is 200 milliliters, so the 250 mark is up around here where there's no graduate. We have a 100 milliliter beaker, same situation, graduates, a 10 milliliter beaker. So these are our beakers. Here we have our flasks. These specifically are called Erlenmeyer flasks. This Erlenmeyer flask here, a 250 milliliter Erlenmeyer flask, has graduates on it. Again, these graduates are not very precise due to their size. The advantage of an Erlenmeyer flask is that you can easily stir the solution that's inside and minimize the possibility of spilling because we have a narrow neck at the top. So the flask has a wider base and a more narrow neck. We can add a stir bar to the flask, which is this small apparatus here. Looks like maybe a little pill. It's a it's a metallic piece of metal that is coated in plastic, right? And so stir bar can be used in your titration lab, for example, to stir your solution with the stir plate, which we'll show you later. Here we have another flask, a smaller 125 milliliter Erlenmeyer flask. These are called graduated cylinders, and as they're graduated like the other two pieces of labware we've discussed, but these graduates are more precise. We see that from 10 to 20 milliliters, we have a large gap, so that we even have markings for every single milliliter. This graduated cylinder is a 100 milliliter graduated cylinder. It holds up to 100 milliliter, and you're going to get a better measurement using this. This is your first degree of precision. Here we have a smaller one, a 10 milliliter graduated cylinder. The cylinder obviously refers to this right here is the cylinder. And they can be plastic and they can be glass. We have pipettes. Pipettes are even more precise at measuring out volumes. These pipettes here are called disposable plastic pipettes. This pipette is a 10 milliliter disposable plastic pipette and can measure out volumes between 1 and 10 milliliters. And you see that between the 1 and the 2, we again have multiple graduates. So we are getting more and more precise, greater capability of measuring out specific volumes of interest. These come in different sizes. Here we have a 5 milliliter plastic disposable pipette. Here we have a 1 milliliter plastic disposable pipette. We always want to work in the smallest volumes as possible to save material, to minimize waste. It's easier on the environment. This one here is a 1 milliliter, so we see that we have the ability of measuring out up to 100 microliters here, 50 microliters there, and even 10 microliter increments on this 1 milliliter plastic pipette. All right, here we have another kind of flask. It's wide at the bottom, small at the top again, so we have a flask. But this flask doesn't have graduates along it. In fact, it only has one graduate right there. It's just one line, and that graduate is very precise for the volume, which is this one, in this case, is 500 milliliter. And when we have a piece of glassware that has just one measurement on it, one graduate, it's called a volumetric flask, or a volumetric glassware. We have volumetric pipettes and volumetric flasks here. So when we're filling up our volumetric flask, we fill it all the way to the top, to this point right here, and we verify that the meniscus, or the slope of the water as it clings to the side of the tube, the bottom of the meniscus should be at that line. So this is our volumetric flask. And we have volumetric flasks of different sizes, all the way down to 1 milliliter volumetric flask. The flasks come with a beveled glass cork or stopper, and you can easily mix your solutions in these as well. So these are very good for making solutions. This other piece of volumetric glassware, again, because it's volumetric, we expect to find one graduate, right? Very thin right here, so we know that it's very accurate. In fact, on these you can find out their accuracy in terms of their error by reading this information up here. This is a 50 milliliter volumetric flask, 50 milliliter volumetric pipette. We have all sorts of sizes. We have a 10 milliliter volumetric pipette, again, just one graduate there, and our smaller one here. Even though we have smaller in the stock room, this is a 5 milliliter graduated 5 milliliter volumetric pipette. And you notice that this 5 milliliter volumetric pipette has two lines, one labeled TD, which stands for to deliver, and one labeled TC, which stands for to contain. So that's due to the fact that in the end, when we transfer water in this, there will be a small amount of water left here after it has been delivered. So if we want to deliver 5 milliliters, we put it up to that line and then we deliver it. A small amount will stay here, which will be equivalent to that difference, and we'll know we delivered 5 milliliters. If we want to contain the 5 milliliters and possibly weigh it or keep it in there for a while, then we move it up to that point. That point is the point that contains 5 milliliters. With both of these different kinds of pipettes, we have a tool to suck water up into them, or suck your solution up into them, right? This little tool is a pump which creates a vacuum by raising this and pulling air in that direction, all right? So we can attach this pipette onto this one. When you do this, you want to be sure you grab it near the end here. If you grab it here and try to place it in there, then you increase your chance of breaking the pipette or stabbing yourself with the pipette or after it's broken. So you want to be nice and close when you do that. When you put the pump onto the pipette, okay? We have a solution here. Then we can draw up the solution. Whoops. There we go. A little bit above, and then we pull it up out of the water and lower it down to the volume that we want. All right, now I know that this is going to deliver 5 milliliters. I can tap gently on the side to remove any, like that, little bit of water on the outside. Okay, when I deliver this, after I deliver, as I mentioned, I can deliver it by taking this off or by just depressing the plunger. But after I deliver it, there is going to be a little bit of water that remains in here, as we mentioned, right? I can tap that last bulb there, last drop there, but this amount of water is not to leave the pipette. It's supposed to stay there. That has delivered 5 milliliters, and they're taking into account that amount there. All right? So that's how we use that one. Our 10 milliliter plastic pipette, disposable plastic pipette. We open up the sleeve and we can place it on there like that. These are generally sterile, so if you're working with something that requires sterility, you'll possibly prepare this way and then wait until right before you're ready to work with it, right? And we remove it, place it in the solution, and we can draw up the desired volume. Again, we usually draw a little past the volume we want. We pull it out, and then we lower it down to the volume we're interested in. And what that does, now I'm right at 1, 2, 3, 4, 5, 6, 7 milliliters. And I can tap off that last bowl, or that last drop at the bottom. If I measure it while this is underwater, if I put this back into water, you can see that that raises up. That volume raises up because of the pressure that this water is putting on the water that's in this cylinder. So that is not a precise measurement. So I have to be sure that I make the measurement with the tip out of the water. And then I can deliver that water where I need to. All right, so there we have our beakers, our flasks, our graduated cylinders, our pipettes with the syringe or the pump. We have our flasks that have one mark, our our volumetric flasks, and we also have our volumetric pipettes.
The volumetric pipette, used to deliver a fixed volume of liquid under specific conditions, using a rubber bulb to draw the liquid into the pipette, and the index finger over the top to control the liquid flow. To use properly, there are 7 steps to follow. Make sure the pipette is clean by rinsing with distilled water. A dirty pipette will have beads of water along the walls. A clean pipette will run smooth and clear. Regardless of the type of pipette, there will be water left in the tip that could contaminate the solution you are pipetting. This is remedied by using the rubber bulb to blow out any water into a waste beaker and wiping the tip after. Pour your solution into a clean, dry beaker, making sure there is enough solution to fill the volume of pipette you are using and to do 3 rinses. Rinsing is done by drawing up a portion of solution and wetting all surfaces by rotating the pipette horizontally, making sure the solution goes above the etched line. Rinsing removes contaminants and water that would dilute your solution. Fill the pipette with solution to approximately 2 centimeters above the etched line. Wipe the barrel with a clean tissue to prevent transfer from the outside the pipette. Hold the pipette vertically over a waste beaker, being held at a 30-degree angle, and slowly drain out the solution until the bottom of the meniscus curve is just touching the top of the etched line. Touch the tip of the pipette to a clean, dry area of the beaker to remove any hanging drops, as they would add extra volume. Setting the meniscus is difficult and requires practice. To deliver the aliquot, place the pipette inside the receiving flask, touching the tip to the glass surface. This allows no chance for loss of liquid. Keeping the pipette vertical and the flask at a 30-degree angle, allow the liquid to drain. Wait 10 seconds after delivery to allow proper drainage. There should be a small amount of liquid left behind in the tip. Do not blow this out. This is extra volume. Take a wash bottle and rinse the walls of the flask to ensure there is no solution adhering to the sides and that it will be mixed with the rest of the sample. Let's review the steps. Make sure the pipette is clean. Blow out any leftover liquid. Rinse the pipette 3 times with your solution. Draw up the aliquot and wipe the tip. Set the meniscus. Deliver the aliquot with the pipette vertical and the flask at a 30-degree angle. Wash the sides of the flask. Now you are ready to pipette.